Now, Shimadzu has recently released some great new technologies with our Nexera XS inert, which is truly a bioinert UHPLC system, as well as our LCMS-9050 QTOF instrument for accelerated discovery. This combination is ideally suited for oligonucleotide analysis. Now, why is this? Just briefly, allow me to introduce you to the front end and the back end of this combination in a bit more detail. Now, starting with the Nexera XS inert. What is a truly bioinert UHPLC system? This is an indication of being biocompatible, which means that it is corrosion resistant to high concentrations of salts and acids, and HPLC molophases such as halogen-containing solvents, thereby making it a versatile UHPLC for such techniques as ion exchange chromatography and side exclusion chromatography. Additionally, it is bioinert, meaning it's metal-free, preventing the absorption of biomolecules to our UHPLC system. Now, starting with corrosion resistance, why do we need this for oligonucleotide analysis? Now, if you look at the structure of oligonucleotides, for example, as you can see here on the right, the phosphate ribose backbone is highly negatively charged, making it an overall polar molecule by nature. Now, to separate such highly polar biomolecules, we usually require very hard chromatographic separation technologies such as size exclusion chromatography, ion exchange chromatography, or most commonly used for oligonucleotide analysis, ion pair reverse phase chromatography. Now I'll be talking about the ion pair reverse phase chromatography because it's the most commonly used chromatographic separation technology for oligonucleotides. Now, in ion pair reverse phase chromatography, we make use of ion pair reagents to interact with the negative charge of the phosphate group of the oligonucleotide, while having at the same time a hydrophobic modality, which allows us to interact with the stationary nonpolar phase of our chromatographic column. Therefore, we can retain our highly polar charged oligonucleotides and separate them on the column. And as you can see here, by increasing the concentration of ion pair reagents, the chromatographic separation of the compound increases all the way up until you can see here that you can nicely separate a thymine 6-mer all the way up to a thymine 30-mer with a range of nucleotide lengths in between them. But ion pair reagent and concentration are not everything. As you can see here, for different lengths of oligonucleotides, we compare the different ion pair reagents and the concentration to see what kind of impact does this have on our MS signal intensity, or basically our sensitivity for our whole system. As you can see, of course, the different types of ion pair reagent, or the concentration, is important, but even more important is the pH of our mobile phase. So you can see by going to a very high pH, by adjusting the pH, we can drastically increase the signal intensity, and therefore the sensitivity of our analysis. So you can see, having a high pH, which again is a very aggressive mobile phase for our system, we increase the signal intensity. Now, if we're changing the pH, it would be obviously very good to online monitor the pH of the mobile phase during our chromatographic separation. And for this, Shimadzu has a great solution with our online pH monitor, which simply can be added to our Nexera XS inert. And this allows you to measure a very wide range of pH in real time and directly store this data together with the chromatograms. Now, being corrosion resistant, of course, means we want to be metal-free as well, to avoid the leaching of any kinds of metal components from the UHPLC system. But this is not everything. Typically, UHPLC systems have about 50% of the analytical flow path with metal surfaces, which are exposed to our analytical compounds. Now, depending upon the chromatographic mobile phase conditions, these metal surfaces will usually be positively charged. Now, if we have biomolecules such as peptides or oligonucleotides, which are negatively charged with phosphates, for example, these biomolecules can very easily interact with the metal surface of the UHPLC system, basically retaining them, absorbing them. meaning we have a loss in sensitivity or even carryover later. So we really want to avoid having too many metal surfaces in our UHPLC system. And we have done this basically by replacing almost all kinds of flow lines with direct PEEK or PEEK line stainless steel pipes to achieve a very high pressure for our UHPLC system. In addition to removing any of the metal surfaces that can interact with our biomolecules from our flow lines, we have also done this with the column, the sample needle, even our PDA and our pH monitor, and have replaced their flow lines with PEEK line material. While as for high pressure components, such as the pump or mixer, we have replaced them with titanium alloys, which are more corrosion resistant. So we are achieving much higher sensitivity. And here we show you some example data. On the left side, if you're looking here at the standard UHPLC configuration, using a standard stainless steel column and looking at mononucleotides, the peak shape and even the sensitivity of the compounds are not very nice. Now, by replacing simply the column with a metal-free bioinert column, you can see there is suddenly detected an additional signal, ATP. But you are also able to see an increase in the signal intensity for ADP. So we are much more sensitive. So when we are switching from a standard UHPLC to our Nexera XS inert, we yet again increase the signal intensity, much more sensitive, and also improving the peak shape. Having this Nexera XS inert be metal-free, we can really increase peak shape and sensitivity for our highly charged oligonucleotide compounds. Now, just to sum up, our Nexera XS inert front end actually is the most competitive bioinert UHPLC system, which is highly corrosion resistant against any high pH concentrations, and also superior metal-free material, which will reduce the absorption of the biomolecules throughout the system. Additionally, we now have an online pH monitor, which really adds value to your UHPLC system and allows us to monitor your pH in real time to be more reproducible. And of course, we have our analytical intelligence of our Nexera UHPLC systems by Shimadzu. Now this is the front end coming to the back end of our mass spectrometer. Just last year, we released our latest LCMS-9050 QTOF instrument for accelerated discovery. This instrument has some great features, such as our trusted mass accuracy, but also now added ultra-stable polarity switching, combined, truly making a versatile and flexible instrument for all kinds of analysis, such as oligonucleotide characterization. Now, what is so unique about our LCMS-9050 QTOF instrument is actually our Reflectron flight tube. This is our patented technology which allows us to have a very high and accurate mass resolution. But what is even more unique is this flight tube is inside a temperature-controlled chamber, the UF flight tube. This allows us to mitigate any changes in length of flight path to environmental temperature changes, or it mitigates any changes in kinetic energy. And this now allows us to have a very robust and stable mass accuracy over a very long period without any need for internal standards, internal calibration, or external calibration required. So you have much more robust measurement and much less downtime. Just to show you some examples of our stable and accurate mass, here we have actually changed the room temperature in the environment by 5 degrees. And you can see in positive and negative mode, we can still achieve a very stable mass accuracy over 24 hours without the need of any additional calibration or internal standard use. Now, of course, with the latest 9050 QTOF instrument, we have an additional unique feature, which is our UF polarity switching. So with our proprietary technology, we are actually able to shorten the polarity switching time in half. Now, for the first time, it is really possible to analyze both positive and negative ions in the same analytical run. So you do not need to segregate and run your sample twice. You can really analyze positive and negative ions in the same analytical run and still achieve high mass accuracy and resolution as you can see here. Overall, this really allows us to do positive and negative switching with high performance on our QTOF instrument, with high resolution and a large mass range, also a very fast acquisition speed, making this the ideal solution for oligonucleotide analysis. So just to sum up, we have this great combination of our bio-inert UHPLC, the Nexera XS inert front end, and of course our LCMS-9050 QTOF back end for high resolution, accurate mass measurements of our oligonucleotides.
Thanks so much for that presentation, Steve. All right, next we'll be going over to Neil Loftus for his presentation, Insights into Oligonucleotide Therapeutics.
Hello, I'm Neil Loftus from the Mass Spectrometry Business Unit overseas. And in this webinar, I will highlight the science of the oligonucleotide therapeutics and identify some of the challenges in data processing. I'm going to explore how can we make things better. As Steve has already presented, our platform for an end-to-end solution for oligonucleotide analysis brings together several enabling technologies. And in this talk, I will focus on two main themes. One will be to provide an overview of the oligonucleotide therapeutics and the challenges in characterization. And the second part is to look at the tools to help accelerate data processing in oligonucleotide analysis. So let me start with an overview of oligonucleotide therapeutics. Oligonucleotides are nucleic acid polymers with the potential to treat or to manage a wide range of different diseases. Although the majority of oligonucleotide therapeutics are focused on gene silencing, splice modulation, and gene activations, there are several possible pathways to expand the therapeutic targets beyond what is generally considered to be a conventional pharmaceutical. The majority of oligonucleotide therapeutics on the market are relatively small biomolecules, which interact with their target molecules via complementary Watson-Crick base pairing. And they work by either enhancing or repressing the expression of target RNA. The chemical structures of 5 single-stranded oligonucleotide drugs that have received regulatory approval from the FDA are shown in this slide. So the question is, how do they work? Two major types of oligonucleotide drugs are currently being developed: the antisense oligonucleotides and short interfering RNAs, the siRNAs. Both share a fundamental principle. An oligonucleotide binds a target RNA through a Watson-Crick base pairing. The resulting duplex directs degradation of the target messenger RNA, mRNA. It's important to note that oligonucleotide therapeutics represent a direct application of genomic information in the designs of drugs. The concept is relatively straightforward. A target RNA is selected, a complementary oligonucleotide sequence is synthesized, gene expression is modulated, and phenotypes are examined. In reality, there are a number of challenges. The key barrier is the inability to deliver the oligonucleotide drug to the target. So there's a number of different processes which are involved in the chemistry to make the difference. Continuing the theme of how do they work, several oligonucleotide therapeutics on the market are single-stranded nucleic acid polymers called antisense oligonucleotides. They're small, 18 to 30 nucleotides of diverse chemistries. Their mechanism of action is to bind to the target mRNA and destroy it by silencing the gene expression process. Gene silencing is a key mode for many oligonucleotide therapeutics currently on the market. The mode of action for antisense oligonucleotides can involve several mechanisms. It can be targeting mutations, it can be targeting the splice sites, or it can target the translation start sites. Each of these mechanisms are designed to provide some mechanism whereby we have gene silencing. And these applications have been used to address a range of different diseases that consider neurological to muscular to metabolic, as well as to developing vaccine. If we look at the current oligonucleotide therapeutics which are on the market, well, 17 of the oligonucleotides have been approved by the FDA and the EMA. 14 therapeutic oligonucleotides approved by the FDA, and 7 of those have been approved by the EMA. If we look at the 3 broad categories of oligonucleotide therapeutics, we have the antisense oligonucleotides. 10 of these have been approved as therapeutics. They're small, 18 to 30 nucleotides, synthetic, single-stranded nucleic acid polymers of diverse chemistries. They bind to complementary nucleic acid sequences, impacting the associated functions of the target nucleic acid. These particular types of oligos have been spearheaded by 2 companies, Ionis and Sarepta. Another type of oligonucleotide therapeutic on a small interfering RNAs. 5 have been approved by the FDA as therapeutics managing rare disease states. They're identified as small double-stranded RNAs that exert their effects by dividing into single strands and binding to the distinct target messenger RNA sequences. And the third group are the aptamers, and 2 have been approved by the FDA as therapeutics. These are relatively short chain nucleotide sequences, generally DNA or RNA of 20 to 100 nucleotides. So what does an oligonucleotide therapeutic look like from a chemistry point of view? In the example here, we have an oligo with a molecular weight around 7 kilodaltons. It has 20 nucleotides, referred to as a 20mer. And the chemical structure of the oligos are characterized by a backbone of nucleic acid polymers of ribose, base, and a modification. Adding a phosphorothioate linkage in one of the non-bridging oxygen atoms, when you replace that with a sulfur, and that approach has been widely used in therapeutic oligonucleotides, by adding the sulfur into the PS linkage, you then generate a chiral center at each modified phosphorus atom with 2 possible stereoisomeric forms. In terms of MS analysis and MS analysis, the bonds in the PS linkages follow predictable fragmentation patterns, A to D, W to Z, with base cleavages. So it's very predictable fragmentation, and it helps us work on sequence information in a far more predictable and accurate manner. There are challenges in developing new therapeutics, and the chemistries are designed to increase resistance to nucleases and improve the target binding affinity. And there's 4 principal ways of doing this. One is through changes to the backbone chemistry. Another one is looking at the ribose modifications. The third one may be considered to be looking at changing the conformation of the DNA analogs. And then another approach would be by looking at alternative chemistries, changes in the nucleobase, for example. All of these are designed to help increase the resistance to the nucleases and try to get it to the target cell and the target site with a greater binding affinity. If we consider now the design considerations in oligonucleotide therapeutics, and as one example, this is a short interfering RNA and an siRNA molecule. It's designed with 2 strands. with one of the strands being complementary to the target transcript. And it guides the Argonaut 2 protein, the AGO2, as part of the RNA-induced silencing complex, RISC, to the complementary target transcripts. The result is cleavage. It's slide-through activity, and that leads to gene silencing. It highlights the changes in developing new synthetic oligonucleotides. And this could be a change on the backbone. By changing this, we're looking at enhancing nuclease resistance and stability. It has a longer circulation and half-life due to the increased protein affinity. However, a higher number of PS linkages leads to in vivo toxicity, so there has to be a balance. It's a design change on the 3' overhang, and that lowers the chance of detection from the RIG1 gene, the retinoic acid gene 1. And there are different alterations to the 2 fluoro modifications and the 2 methyl modifications. Both are designed to look at binding affinities and looking at reducing off-target toxicity. And finally, by coupling with the GLACNAC, that can accelerate the hepatocyte uptake with a high specificity only by the liver. So there are many different design considerations when looking at an oligonucleotide therapeutic. Creating or modifying oligonucleotides is complex. Synthesis cycles supports 4 main chemical reactions, and that cycle is repeated until the required nucleotide sequence has been achieved. The challenge is, by repeating the cycle a number of times, the greater the number of impurities, which leads to a higher data processing time. So it's complicated. The more the cycle is repeated, there is a higher risk of amplifying impurities. So within data processing, we have several challenges. We need to think of a way of checking a full-length product verification for its molecular weight. We need to check its sequence. And then we need to look at an in-depth analysis and characterization of the impurities. So let me walk you through how we would do a full-length product verification workflow. We'd open the software, we'd open a batch of samples, and in this instance, we'll just actually put the sequence as a text. So we'll just copy and paste into the field. The other thing I'd like to point out is that we've got shortcuts, and in shortcuts, we've just got 4 particular icons, which enables very, very straightforward workflow. Once the sequence has been entered, the next step is to click on Identify. This is a fully optimized processing. It's quick, it works with pre-optimized defaults. By clicking Identify, it starts a cascade of processing events, which includes peak detection from the LC data streams or the MS data streams. It considers automated deconvolution with predefined optimized defaults. It searches for all of the selected modifications, checks for impurities, merges spectrum to optimize the sequence coverage, and quite a bit more as well, actually. The result in the screenshot shows the molecular weight and sequence is in agreement with the expected product. A very easy to work with data processing environment. So to look for a full-length product verification, we can just simply put the sequence and ask the software to actually process on that basis. However, for more advanced needs, we have the settings editor. The settings editor is a powerful and flexible toolset. In the sequence settings, the editor shows the chemical structure for the synthesized oligonucleotides, with options to show the MS fragment bonds and a table to highlight base, the modified base, the linker, and the ribose, with the corresponding formula and monoisotopic mass shown. It's a fully editable table, and users can change each field to meet workflow needs. And to create further flexibility in the search list, it can be modified to meet differing workflow needs. The users can add or remove different nucleobases, can add or change different linkers, can add or change different ribose units and base modifications. And the settings also include different ion additions and changes to the modification settings. So it can be expanded, it can be scaled, it can be changed according to particular needs. In the settings editor, we have options for choosing how the data processing is driven, whether it is LC or MS driven, and they're both supported by individual peak detection parameters. In the example shown here, the data processing is LC driven using either UV or PDA data streams, and the relative peak area thresholding is calculated based on the full-length product being the most dominant peak in the chromatogram with these settings. Other parameters are available to change but are typically set to default, but it does include the MS deconvolution spectrum settings. There's an opportunity to change the unknown modifications, the MS and MS spectrum, and calculating relative abundances used in the impurity thresholding. As peak integration is a critical step when we're looking at impurity reporting, the LC and MS data streams need peak integration parameters which are specific to each data stream. As the LC was selected as the primary data stream for data processing in the previous screen, this dialog now provides options for data integration, which also includes advanced settings for time-specific integration changes as well. It's a very flexible environment, very easy to work with, and it's the same that we have in our normal chromatography. The settings editor provides a really powerful and flexible environment for different needs. But once it's set up, then users just simply need to click on Identify and process the data ready for review. So Insight Biologics supports several tool sets for data review. In its simplest form, to share an extracted mass chromatogram for the full-length product with the corresponding sequence and mass error. And here we have a chromatogram, and then we have a table. The results table can be filtered, only showing the full-length product verification, and all of these objects that you see, whether it's chromatogram, spectrum, whether it's a table, they're all dockable panes, which really does allow for a fully customized data review experience. So flexibility in the layout is really important for us. Insight Biologics can display the chromatogram spectrum data and review results table. But a core need really is to actually check the sequence coverage. And by clicking on the coverage icon, users can make sure the sequence is correct. In this example, the sequence coverage is complete and it's in agreement with the expected sequence. The sequence coverage is calculated using the MS fragment table. If there was a change in the modification or a change in the sequence, then it will be highlighted in this screen. And there's two ways of looking at it. This is the branch mode, and then the next slide, it will be actually the fill mode. The fill mode shown here is the same data content as was shown previously in the branch mode. It's just a different visual experience. And the fill mode will display the fragment cover map, which are linked to the MS spectrum. It's allowing a really quick way to check the sequence is correct. And as with the branch mode, it will identify any incomplete or missing coverage. It'll identify where the modifications are. And it will show where possible longmers or shortmers are as well. So a very simple visual experience. As Insight Biologics has been designed with dockable panes, it does present a very flexible tool set for reviewing data. And this is one example of bringing it all together in one easy to visualize data presentation. And here we show the LC chromatograms, looking at the LC PDA, we've got the LCMS, we've also got the spectrum, we've got the deconvoluted MS spectrum, we've then got the MS spectrum, together with some tables. And it's important to note that inside the tables, users can change which fields are being displayed. They can then filter on each one of those fields to display the information that they want to. So it's a very flexible environment, easy to use, and it makes the whole experience of working with the data very straightforward indeed. And this is another example of looking at the data view options. Again, we've got a chromatogram, we've got some results table, but in this view, we've also included the branch mode for looking at the sequence coverage. So users can actually look at whether the full-length product has been made, make sure the sequence is correct, and check for the data quality. One of the key design elements in terms of data processing software is to have a great deal of power in what it can do, but also have the flexibility of addressing different needs. And one of the key areas which is still evolving is really about the flexibility for reporting impurities. And robustly reporting impurities in oligonucleotide analysis is challenging. The paper published by Rental et al. Highlights the key issues. Oligonucleotide therapeutics have got a molecular weight typically between 6 and 9 kilodaltons. There is a presence of a large number of impurities, and they're at low intensities. And those impurities are also structurally similar to each other and to the parent product. It also highlights the challenge in meeting regulatory needs, which are well defined for small molecules, but still evolving for oligonucleotides. So, oligonucleotide therapeutics are explicitly excluded from the scope of the ICH Q6A. But there's general concepts in the quality guidelines that are currently being considered and applied to oligonucleotide manufacturing, with the most relevant being ICH M7, Q3C, Q3D, Q7, Q11. One thing that's very notable is the diversion from the guidance when it relates to the ICH Q3A, which is about impurities in new drug substances. While the general concepts of reporting limits, identification limits, and quantification limits still apply, higher thresholds have been accepted for oligonucleotides than for small molecules to date. So there needs to be some flexibility in how we do this. Insight Biologics can approach impurity reporting by considering an LC-driven workflow. For each detected LC peak, and in this context it can be either UV or PDA data streams, the LC peak is then aligned with the MS data streams, and the process for all MS signals from the LC peak start to the LC peak end. All ion signals in the TOF mass spec scan and DDA and SMS scans are processed against an oligonucleotide sequence, including selected target modifications, ion additions, sequence deletions, the short MES, and the sequence additions, the long MES, and a dictionary of predictable impurities. So for every LC peak start and peak end, the mass scan is checked against the oligonucleotide target list, sequence modifications. Multiple extracted ion chromatograms are then used to calculate the relative ion abundance of each detected impurity. And in this example, the approach was applied to the analysis of a 20-mer oligonucleotide separated using ion pair on the Shimadzu inert LC system. By setting the PDA threshold to a usually defined reporting level, in this example 0.1% of the dominant LC component, for each detected PDA peak, MS scans are checked against the oligonucleotide target list, including the sequence modifications. Filtering by the relative LC percentage peak area threshold can then be applied in the settings method, or simply by filtering the results table. So this gives you the flexibility to look for different impurity needs depending on the process at the time. Once the data has been acquired, the next step is to report the data. And inside Insight Biologics, 4 report templates have already been supported, enabling different report options to be presented. It simplifies reporting, makes it easy, but gives the flexibility to report the parameters and values that are required for that workflow. One of the key goals within Insight Biologics is to drive high data quality. It's a primary objective from the inert LC system to the high resolution MS platforms. And one of the key areas that we've added value is working with a patented algorithm for merging DDA, MS, and S spectra. One of the challenges about just using an average DDA and SMS spectrum is that it is inherently biased to charge states with a higher signal intensity, and that results in a weighted MS/MS spectra. Insight Biologics considers this in a very different way. It looks at every fragment ion in every DDA MS/MS mass scan for all charge states, and it registers the most intense value into a merged spectrum. The bottom line is, this new approach delivers better data quality, and it helps with sequence coverage. It's adding value to the whole process. So, to conclude, we've got the tools to deliver a meaningful impact on oligonucleotide characterization. We're delivering an end-to-end solution for full-length product verification and sequence confirmation, with the flexibility to work with different approaches in impurity reporting. We're driving the higher data quality and data acquisition with inert LC systems, inert LC core materials. We've then got the LCMS-9050 QTOF, which has got a very high sensitivity, high mass stability, and then we've also got a different user experience. We're looking at a better user experience for data processing. So Insight Biologics has been designed to be quick, it's been designed to be easy to work with, it's been designed to be really very flexible indeed. It's also got the tools to adapt to new challenges. It can work on focused workflow software, and it's in a compliant-ready software application. And with that, I thank you for your attention, and the questions and answers will follow.